Good morning, everyone. COVID-19 conversations are brought to you by the African Alliance with support from the South African Medical Research Council and the Department of Science and Innovation and the Vaccine Advocacy Resource Group, which is 100% funded by activists. We bring you these calls in partnership with the Community Constituency COVID-19 Front, the Treatment Action Campaign, and APA. Thank you for joining us. These calls take place every Thursday at 10 a.m. and seek to bridge the divide between civil society and those who hold power, influence, and platform in the COVID-19 research space. All of our recordings, along with the transcript and the Q&A sessions, can be found at africanalliance.org.za. We encourage pre-submitted questions and welcome questions or comments in the chat box during the call, and we will do our best to answer all of them. Those we do not get to, we will reach out to our guest after the call to secure those answers, and those will be included in the transcript. So welcome. Today we speak to Professor Graeme Meintjes, who is an infectious diseases physician and the second chair and deputy head of the Department of Medicine at the University of Cape Town. He also holds the South African Research Chairs Initiative Chair of Poverty-Related Infections. Over the last year, he and colleagues at Groote Schuur Hospital have been managing patients with COVID-19 admitted to the wards of the hospital. We also speak to Dr. Amy Ward, who is the site director of the UCT Khayelitsha Clinical Research Site and a qualified medical doctor who has worked in the community of Khayelitsha for 8 years. She's an experienced clinical trialist focusing on HIV and TB and more recently COVID-19. She's currently leading the Khayelitsha CRS team as Principal Investigator on 2 COVID-19 vaccine trials and sits as National PI for one of the world's largest COVID-19 natural history observational cohorts studies. So colleagues, today we really want to speak about HIV. We've heard of this vector, we've heard of some of the news coverage in an article that was released a few weeks ago. 4 of the world's preeminent scientists and researchers shared their views. They packaged it as a cautionary tale around the use of Ad5 in the context of COVID-19 based on our collective history in HIV vaccine research studies. Today, we want to ask Professor Graeme Meintjes and Dr. Amy Ward to take us through some of the basics of the phase 1 trial that is being launched in South Africa, in Khayelitsha in particular, amongst other sites. And from there, we hope to engage in a robust question and answer session, as well as a review of some of the commentary that has come out from global and national leaders around the use of this particular vector. So again, Professor Graeme Meintjes and Dr. Amy Ward, welcome. We will hand over to you now to give us an overview of the trial in the next 20 minutes and looking forward to the engagement. Thank you again for making the time.
Thanks so much, Tian. You can hear me?
Yes, we can hear you and we can see you.
Okay, great. And thanks for the opportunity to discuss this trial with you and colleagues in the African Alliance this morning. So I'm going to just provide some background regarding the science that has led up to this study. And then Amy's going to discuss the design and conduct of the study that we're planning. So if I can just share my presentation. So hopefully it's coming up there.
Yes,
and just put it on slide view. So what I want to discuss with you today is a phase 1 trial. It's a fairly small, early in development study that we are conducting of a second generation SARS-CoV-2 vaccine that includes both the N and the S protein, and I'll come to that in more detail, with, as you say, an adeno5 virus vector. Just by way of background, I don't want to go into great detail about the vaccines that we've been hearing a lot about in the news over the last 2 weeks, but the existing vaccines that are coming into clinical use, and we know that there's 6 or 7 of them across the globe that have come through phase 3 trials and are now being incorporated into programs, have potential deficiencies. And I've really underlined the word potential because these are question marks, and only with the course of time will we be able to address whether these are deficiencies or not. The potential deficiencies are that are in large part related to the fact that these vaccines just incorporate the S protein of SARS-CoV-2 and expose the immune system to the S protein, which is obviously just a single component of the vaccine, And if that S protein mutates, as we've been seeing, for instance, with the mutation that has arisen in South Africa, then the efficacy of the vaccine might be compromised. So the potential activity of vaccines against variants with mutations in the S protein is a big question mark that could reduce efficacy and even make the vaccines ineffective if there are multiple mutations. The other question mark, and this is something that we really don't know, is how long the immunity induced by the current vaccines will be. That tend to focus on S protein responses and drive antibody responses. Although there are T cell responses, the main focus has been on generating antibody responses to block entry into the cells, the human cells, and effectively achieve neutralization. And so, you know, that's a question mark. And if the long-term protection is not provided, we are potentially going to need booster doses or more effective vaccines. And the other question mark over the current vaccines is maybe they're effective at preventing disease and stopping people from being admitted to hospital, preventing death. But how effective are they going to be at preventing mild disease and asymptomatic disease, and therefore ongoing transmission, and therefore achieving what has been discussed as the herd immunity or the community immunity that we're aiming for? Because if the vaccines are not effective at preventing transmission, then then they really can't achieve community immunity. It's really just about individual immunity that can be achieved through that. So all of these issues and potential deficiencies mean that we can't sort of arrest and say that we've achieved, you know, 6 or 7 vaccines and those will help us to address the epidemic. We need to look for next generation vaccines that could address potentially these deficiencies. So I just want to go through some background before I delve into the details. But this is showing the SARS-CoV-2 virus, and on the surface you can see the spike protein or the S protein, which is what most of these vaccines that we have contain. But in the middle, you can see the nuclear capsid, or what's called here the nuclear protein, which is the blue structure, which is found within the virus. Now, this novel vaccine that we will be testing has been developed by a biotech company in the US, Immunity Bio, and it has a vector, and I'll come back to what a vector is, but a vector is essentially another virus, and in this case, the human adenovirus, which is a virus that causes the common cold that many of us have been exposed to. But this vector in the vaccine has had several genes taken out of it to make it very unable to replicate effectively in a human body. And the vector that we're going to be using has had additional deletions compared to the ones that have been used in previous trials, such as the HIV trials, and therefore remains relatively immune silent. And there is evidence to support that statement. The vector was initially developed by Immunity Bio for cancer vaccines and has been used in phase 1 and 2 trials in cancer vaccine trials in over 150 participants in the US. And the important thing about this vaccine, as opposed to the ones that we currently have, is it doesn't just contain spike, it also contains the nuclear capsid protein. So it's both S and N. And the nuclear capsid is linked to a genetic segment which allows the nuclear capsid protein to be more effectively processed within infected cells to present to T cells and generate T cell immunity. So this just demonstrates what I said in the previous slide. Essentially, what it's showing here is a SARS-CoV-2 virus. And what's taken out is the genes for the spike protein and the nuclear capsid protein. And they are then put into the vector. And this is what a vector is. A vector is essentially a hollowed out adenovirus. Several genes are taken out of it. And then you insert the genes from SARS-CoV-2 that you want to have to provoke an immune response when the vaccine is given. So the vector is like a vehicle, but it's a very weakened vehicle in that it's not able to replicate and cause disease as an adenovirus would. And with this vector, compared to previous vectors used in the HIV vaccines, there have been additional genetic deletions to reduce immune responses to the adeno5 virus. So this is just really showing the concepts again. You're taking an adeno5 virus, deleting 3 genes compared to 2 genes, which was the case in the previous vector. So in the previous vector, E1 and E3 were deleted. Here, E2B is also deleted. And you're taking DNA segments for the S and the N protein and inserting them in the vector. And this is what essentially the genetic makeup of the vaccine looks like. You've got the S and the N with the enhanced T cell signaling domain and additional genetic sequences to allow it to function as a vaccine when it's delivered in studies and potentially in more broader use. So this is a second generation human adeno5 serotype 5 vector with 4 deletions. So the E1 and the E3, and then 2 deletions to achieve deletion of the E2B gene. And essentially, that makes it far more immunologically silent, which is important, and I'll come back to that, far less likely to provoke adeno5 virus responses in humans. And the way that what this is just showing is that with the previous vector, there was this adenoviral fiber, which is a protein which provokes a lot of the immune response, was very strongly present in this Western blot. Whereas with this new vector, you see very little of this protein being produced. This means that if you take people who previously had adeno5 infections, you can vaccinate them with this vaccine and achieve vaccine take. You can also vaccinate people in the cancer studies up to 5 or 6 times a year without the adeno5 virus immunity preventing further vaccine take. And this is just indirect evidence that there's less immunological response to this vector than the previous vector. So this is a detailed slide, but I'll just summarize that what this vaccine is attempting to achieve is to generate both antibody responses to S and N, but also achieve very strong T cell responses, both CD4 and CD8 T cell responses to both S and N, and to therefore both block the virus from getting into cells, but also to effectively kill cells that are infected, for instance, in the nasal mucous membrane. So what are the studies that have been done to date? This is a study that is available as a preprint in bioRxiv. It's being written up for submission to Science. This was a study done in rhesus macaques where they evaluated this vaccine with a prime subcutaneous injection and then an oral capsule which bypassed the acid of the stomach and was intended to achieve immune responses in the gut. And I'm not going to go into detail about this, but you can see these are the anti-spike IgG, so the antibodies responses to spike. Blue was those monkeys who received placebo. Red is those who received the active vaccine. And you can see by 6 weeks or so, all of those that received the active vaccine were generating IgG antibodies. In terms of neutralization, ability to prevent the virus from getting into cells was achieved in all but one of the vaccinated monkeys. Then in terms of T cell responses, in blue, the monkeys who received a placebo, in red, those who received the active vaccine, and looking at the T cells' ability to produce interferon gamma, which is a protein when they're stimulated, that they produce that. And you can see that that was produced in response in the lab in response to S protein as well as in response to N protein. So in summary, you're getting antibody responses as well as T cell responses. This is very exciting data from the same experiments in the monkeys where, and I haven't seen directly comparable data for the other vaccines, where you're looking at the amount of virus in the nasal passage after a challenge with virus at 7 to 8 weeks after the initial vaccination. And this is showing the subgenomic RNA, and you can see that in blue, it does decline in placebo recipients, but a far more rapid decline, and all cleared by 5 days in those who received the vaccine. So it suggests that this vaccine is able to generate immune responses that rapidly clear the virus from the nasal passages, which could, and I'm using words such as could and potentially, because we will only know this when we do larger human studies, could potentially help in preventing transmission, because reducing carriage of the virus. So then, just to say that there has been a phase 1 study done of 20 participants in the US, it was actually just a press release released yesterday to say that there were no safety concerns in the 20 participants, and that there were good T cell and antibody responses generated. The details are still to come out in preprint. The FDA has also issued a license for the vaccine to be developed not just in an injectable form, but also in a sublingual capsule. And that would be something that people put under their tongue and then gets trafficked into the tonsillar region and generates immune responses there, as well as the oral capsule that I referred to that bypasses the stomach acid and generates immune responses in the gut. And that's very exciting, because in a lot of infectious disease fields now, people are looking at how do we generate mucosal immune responses rather than just systemic at the site of entry of viruses or other infections. And the data from the animal studies is promising. It's now going to be looked at initially in studies in the US in humans as a booster with either sublingual or the oral capsule. And we would look to partake in those as well. To move on to the issue of the adeno5 virus vector, as Tian referred to, there has been concern over use of this for SARS-CoV-2 vaccines. The basis for this is 2 trials that were done over 10 years ago. The STEP trial done in the US, which was an HIV-1 vaccine trial using an adeno5 virus vector developed by Merck. When they looked at people who were adeno5 virus negative at baseline, there was no difference in HIV infection, so the vaccine didn't work. But in people, and this was specifically in men who were adeno5 virus positive at baseline, or in uncircumcised men, there was a significant increase in the risk of HIV infection. You can see per year 5% versus 2%, 5% versus 1.5%. But the incidence of HIV was similar in Ad5 seronegative men and circumcised men. And this is just showing you the data for men who received vaccine versus placebo. If they were Ad5 positive, it was 29 infections versus 13. If they were uncircumcised, 20 versus 6. So, small numbers, but there was a significant difference. The Pambile study that was using the same vaccine construct in South Africa was then stopped because of the findings of the STEP trial. In the initial results, there was no difference in HIV infections between vaccine and placebo recipients. But when they followed these participants out further and called back some for further follow-up, there was an increased risk of HIV infection, so a hazard ratio of 1.5. 82 infections in the vaccine group, 54 in the placebo group. There was a significant increase for men, but not for women. And this is just showing the data here that I mentioned. So compared to in men, compared to those who received the vaccine versus placebo, it was 3.9% versus 1.4%. So again, relatively small numbers, but a significant difference. So what was the potential mechanism? This was studied in immunology studies and animal studies, and was thought to be related to the fact that this vector, the adeno5 virus vector that was being used for this HIV-1 vaccine, was activating target cells, so the immune cells in the genital mucosa that are target cells for HIV, and therefore potentially increasing risk. And as Tian mentioned, this has been raised as an issue by some eminent scientists in the US, a cautionary tale about using adenovirus vectors for SARS-CoV-2 vaccines. We've obviously grappled with this issue over the past few months together with colleagues at the Medical Research Council. And the important issues about how we're addressing this, firstly, is in the design of this vaccine. This is, as I've tried to emphasize, a next generation adenovirus vector that has had additional genetic deletions. So the E2B gene, which results in the production of a lot of the proteins of adenovirus that generate the immune response. And this evidence of the fact that this reduces adenovirus immune responses is the fact that you can give multiple vaccinations to individuals. So given that the mechanism of the increased HIV risk was thought to be the adenovirus immune responses, the fact that this is reducing those responses would lead us to conclude that it's less likely that we will see this risk with this vector. However, obviously, we need to take caution, as was suggested in that article, and it's been addressed in the trial design as well as the informed consent. And Amy's going to speak to that in more detail in her presentation. Just to say that we will be doing this trial at our Site B clinical research site. We've had experience, this team, this was the team that started out doing the HVTN 702 trial 5 years ago. Many of the members have changed, and the team has grown in size to about 24 people. We were a very successful site as part of the HVTN 702 trial. Unfortunately, the results of that didn't show success, and that was not an effective vaccine. This team has also been involved in conducting the J&J Ensemble trial and recruiting participants there. So very experienced in vaccine research and have built up strong collaborations with community organizations and the CAG in Khayelitsha, and will be led, the trial will be led by Amy with my full support and participation. So just in summary then, this is a novel vaccine incorporating N and S. The other vaccines that we have just focus on S, targets enhanced T-cell responses, and that potentially, as I said, this is why we do studies, is to prove or disprove this, could potentially provide immunity against variants, immunity that is of longer generation because of the T-cell responses, and more effectively prevent transmission by clearing the infection faster. Adenovirus vectors were previously associated with increased HIV risk in the STEP and Pambile trials. The vector in this vaccine has been further modified to reduce immune responses and therefore we think likely reduce the risk of HIV infection related to this. Nonetheless, this is being addressed in trial design and informed consent. This is a phase 1 trial. If we show benefit like has been shown in the US phase 1 trial, the plan is to move rapidly to phase 2-3 trials, and including the oral and the sublingual boost, which are stable at room temperature, which is a very important feature. And the company is committed to partnerships in South Africa and established partnerships already with BioVac in terms of manufacture and the Medical Research Council. So what I've tried to present to you today is that, with most things in medicine and developments, there are potential pros and potential cons and potential benefits and harms, and we need to weigh those things. We've been thought very carefully of this. We think there's huge potential benefit from this vaccine. There are potential concerns, and we've tried to address those carefully in the design and our consideration of whether we participate in this trial. And I'm going to hand over to Amy to give more details of the trial.
Hi, thanks, Prof. So I'm going to take you through now. I'm just going to share my slides. Can you guys all see that?
Yes, the slides are clear. Thanks, Amy.
Okay, great. So I'm going to take you through now just a brief overview of the study that we have planned using the vaccine that Prof. Meintjes has just described to you and given you a background on. So we've named this study ProViva SA1. And let's have a look. The full title is a phase 1B open label study of the safety, reactogenicity, and immunogenicity of a prophylactic COVID-19 vaccination using a second generation E1, E2B, E3 deleted adenoviral platform in healthy South African adults. So just to package out break down what that big package means. So first of all, we are being sponsored by the Immunity Bio Inc., which is the biotech company Prof introduced you to. This is a phase 1B study. So we're not looking at efficacy. We're looking at early phase endpoints, mainly safety, but also immunogenicity. It's an open label study, so there is no blinding. Participants are aware of what they are getting. There's also no randomization, as there's no control arm. So all participants will be getting the study vaccine. The study product that we are testing, the full name is HAD5 S-Fusion plus NETSD, which Prof has described to you. It's given in this protocol by a subcutaneous injection in the prime boost strategy with a prime at day 1 and a boost at day 22. We are looking to enroll 35 participants in total, and the target group of participants are 18 to 50-year-old adults who are otherwise healthy and don't have comorbidities. If we look at the study objectives, firstly, we are looking at, is this vaccine safe to give? And is it tolerable? So what are its reactogenicity, what is the reactogenicity profile? We'll be looking at all safety events through 6 months after the last study dose. We'll be looking at adverse events, and then obviously the tolerability of the vaccination includes the usual symptoms that people are expected to get after vaccination, just to check that these are not out of keeping with what is normal for vaccination reactions. Under safety, we are also monitoring for HIV infections, and we'll be looking at this through 12 months post the last dose of study vaccine. And this links up to what Prof has been saying about the vector in this trial. We want to be extra careful that we are taking note of HIV infections, even though we feel like the data is supporting that the risk is not going to be there. We are definitely going to monitor for it, so that should we see a signal that this risk is here, we can act on it quickly. We will also be looking at immunogenicity through 12 months post last study injection. And as Prof explained, we're looking at both the antibody and the T-cell responses. And importantly, to back up the rationale around the vector mechanisms, or the old generation vector mechanisms of the increased HIV risk, we'll be looking at the immune response in participants to the Ad5 vector itself at baseline, at day 29, and at 6 months, so that we can confirm what we've seen in the previous trials, that this vector does not actually induce an immune response from the human immune system. So the study schema will be taking healthy volunteers and putting them through a screening process. If they are found eligible to enroll, they'll be enrolled in a stepwise fashion, starting off with cohort 1 of 10 participants. We will be giving a lower dose in this cohort, and we will then pause for a safety period just to check that there haven't been any major safety concerns at the lower dose. The safety period is 7 days post the first dose of injection. If we clear those 7 days without major safety concerns, we can then expand into cohort 2, where we will be testing the higher dose, which we think will get the best immune response. And again, pause for 7 days post first dose to ensure that this higher dose has no major safety concerns. Should this prove true, we will then expand to enroll the entire cohort to total 35 participants at the higher dose. If we are concerned about safety profile in the higher dose, we will enroll the expansion cohort at the lower dose. Just a very brief overview of the study procedures. I wanted to highlight in the slide when we get injections, when we're doing safety assessments, and when we're specifically looking at HIV mitigation strategies. So as you can see, the study takes place over just over 1 year. So it's actually 1 year post the second dose, taking us to day 387, where trial participants will exit the study. They are given a prime at day 1 and followed up closely in that first period at day 8 and 15 for safety endpoints, just to check that we're not giving something that is unsafe in humans. Then they will also get a dose at day 22, so that will be the boost dose. And again, they are followed up very closely in this period immediately following the dose to check for safety endpoints. Then they will go into an extended follow-up phase where they'll be followed for safety through 6 months post the last dose and immunogenicity right up until the end of the study. Importantly, on top, I've indicated where we start implementing our HIV risk mitigation strategies. So you can see from the very beginning at screening, we already start implementing our HIV risk mitigation strategies, and it is done regularly at basically every in-person visit throughout the study. And I just wanted to highlight here that Khayelitsha CRS, the site that I lead out here for UCT, we are an HIV prevention research site. We have taken upon COVID trials now as part of the global efforts to aid in the pandemic. But this is what we are used to. We know how to counsel pre and post-test. We know HIV prevention strategies, and we know risk reduction counseling. So we are very comfortable with overlaying these risk mitigation strategies onto a COVID-19 vaccination protocol. So our safety assessments, just to go over in brief, we'll be doing full symptom inquiries, physical exams, vitals, hematology and chemistry panels, urinalysis, pregnancy testing, diary cards post-vaccination, as well as telephone and in-person follow-up of all safety events. Our HIV risk mitigation strategies. So as I said, we start at screening prior to study enrollment, and we are explicit in the informed consent. I'll give you a little snapshot of the language, where we explain to participants that although this vaccine does not have anything to do with HIV prevention, that the vector that is used in this vaccine, while similar to a vector that was used in an HIV prevention study, is not the same. Nevertheless, we have taken extra care to design the study with additional procedures to try and educate people and make them aware of the potential HIV risk that may be there, although we really think that the data is pointing that the risk is not going to be that high. So in the informed consent, we start off with explicit language and go through with each participant before they agree to start the study. In the eligibility criteria, we have added specific criteria to only include men at low risk of HIV infection. And you know, this is something that we did in response to really just engaging with ethics committees and the data on this. And this is a phase 1 trial, so we should just be looking at the isolated immune response and at not in people with any risk of any additional side effects. So we're actually going to exclude men who do not screen low risk for HIV. And we also implement risk reduction counseling for HIV from the very beginning. While on study, participants get 3 monthly HIV testing, obviously paired up with pre and post-test counseling by our seasoned HIV counselors, and a regular HIV risk reduction counseling while on trial. And our HIV risk reduction package includes a behavioral risk identification. We discuss biomedical and non-biomedical proven HIV prevention methods, including PrEP, medical male circumcision, male and female condoms. And if participants are interested in PrEP or medical male circumcision, we will ensure that participants are linked with this care and are, as a site, quite experienced in PrEP provision through our HIV prevention work. So in the informed consent, this is the language that we have included that's gone through the ethics committees to just really highlight to people that this is a possibility and that they should be comfortable with this language and have a good understanding of what we're talking about when we talk about potential for increased HIV infection. I'm not going to go through it all, but in short, we make it clear that this vaccine is not testing if it can prevent HIV. This is a COVID vaccine, that in 2 prior HIV vaccine trials, that use an Ad5 vector that is similar but not the same to the vector being used in the study, showed increased risk in men, in particular groups of men. And that because of this, we will be doing additional procedures going during the study to ensure that the risk of HIV in our participants is incredibly low, or is lowered as much as possible. Immunology assessments in the study. So we'll be looking at humoral immunity, which is our antibodies, and also cellular immunity, the T cell functions, as Prof has gone over. And basically, this is just some pictures showing the ELISpot and the flow cytometry to show how we look at what cells do pre and post vaccination. And that is the brief overview of the study. It was quite quick, but I'm happy to take any questions.
Thank you so much, both Prof. Meintjes and Dr. Ward. I think you have preempted a lot of the questions that came in advance. The questions we're really focusing on, because of the history, unpacking the informed consent and what that means, and thank you for touching on that, but also looking at what is this basket of HIV prevention options that we are offering the men in this trial going to look like. But some interesting reflections that are coming through is, you spoke about this US trial. What is the difference between that phase 1 trial and the one that you're attempting to conduct here? And could you specifically comment on rates of circumcision between the two groups, as well as HIV prevalence between the group that was part of phase 1 in the US and the potential group that will be enrolled here in South Africa?
Yeah, thanks, Tian. I've seen Tando ask that question. Thanks for the question, Tando. And it's not the first time I've been asked. It's an important question. Why, if a phase 1 trial was done, is being done in the US, is it also being done here? Firstly, to say that, just to answer the one question about the participant numbers, the US trial was initially designed exactly the same way, to enroll 35 participants. They enrolled 20. And then they put in an amendment to not enroll an additional 15, but rather to start with the oral and the sublingual. And so they're very quickly going over to that. And I would say that it's quite likely that we might do the same, is enroll the first 2 cohorts here, and then get in an amendment to look at the sublingual and the oral capsule as an alternative strategy. One of the reasons why you can see that in the US, they're going to be, there are various different arms that one can test. You can give a subcut and then 2 oral boosts, or try an oral and then an oral boost, and etc. And subcut and then sublingual boost. And what we're planning is to do the same foundational study here with the 2 subcuts as been done in the US, and then to do different, provided we show the similar responses, to do different arms here compared to the US. And the totality of the data will then tell us what to take forward to phase 2 and 3. And we're hoping that we will be able to take forward a strategy, for instance, a single subcut injection with 2 oral tablet boosts. But we want to compare multiple arms between the US and South Africa of different strategies, and find an effective one, but that would also be one that is most easily to roll out. So I hope that addresses the question of the numbers and how these 2 trials are going to work in parallel. Just to say, in terms of why South Africa, why are we also doing it in South Africa from the start? We wanted, we're aware that T cell responses can have a genetic influence. And therefore, we wanted to see that, to ensure that the T cell responses that are being seen in the US study are going to be seen similar in our population. So there are reasons why we wouldn't necessarily expect to see the exact same responses in a US study and a South African study. There's also differences in co-infections in the microbiome that could lead to different responses in different settings. And therefore, we when we got involved, we were keen to start the development process, to partner and start the development process, be involved in the development process in South Africa in parallel with the US, and start with those foundational phase 1 studies. But as I said, what we are looking at is doing the first 20 participants similar, and then they would be done differently, in the US and South Africa. So I hope that addresses that question.
Yeah, Graeme, could you speak a bit more around the circumcision status of the 2 groups?
Yeah, so I'm not aware of the data for the US on circumcision. And I think that the population that would have been enrolled in the US would have been a low-risk population for HIV. So I don't think that the US, either this phase 1 study or any further development in the US is going to answer the question that we need to answer in South Africa about HIV risk. I think it will only be answered in South Africa. And you know, I think the risk of HIV is going to be very low in the US. I mean, the other thing that one must remember is those HIV studies that I referred to, both the STEP and the PAMBILE, those studies went out of their way to enroll people at high risk of HIV. Because you know, that was what they were testing. Do you prevent people at high risk of HIV from infection? You know, this is going to be a far broader study, and in a way, as Amy said, we are, you know, trying to preferentially, at least in the early phase, avoid putting people at any potential risk, even though we think it's lowered, and therefore avoiding avoiding participants with HIV. Obviously, it will lead to a lot of challenges as we go forward in the development process, because at the same time as wanting to avoid any risk, we also want to answer the question, is there an increased risk? Our hypothesis is that there is not an increased risk with this vector, but it's only through research and doing it with people having full knowledge and having all of the HIV prevention strategies in place that we will be able to address that question in the trial.
Absolutely, thanks for that. I think, you know, if one thing that we've learned since the advent of COVID has certainly been forefront of our minds is that context is everything. The science is constantly changing. And what was particularly interesting for us to observe is having almost two narratives around the use of Ad5. I mean, you had Larry Corey saying that if he was a sub-Saharan African country and there was a choice, he wouldn't and can't see why, you know, we would go for an Ad5 vaccine when the alternative choices. Then almost at polar opposites to that, you had Professor Glenda Gray from the MRC speaking about the trauma of PAMBILI, but also about how important it is not to throw the baby out with the bathwater and to be really, really diligent in terms of how we move forward, because of course, we all want answers to these kind of questions, which brings me to the point of our national regulator.
Sorry?
Which brings me to the point of SAPRA, the regulator. Could you give us a flavor and a feel of what some of their concerns were and how you responded to those concerns?
So just before I go there, you know, I think you know, when the article was written regarding Ad5 and that subsequent discussion, I would add the caveat that they weren't discussing the issue of modified Ad5 vectors. They were just, you know, saying we shouldn't be using that same Ad5 vector. The question of whether modified Ad5 vectors are going to adjust that risk is obviously still something that needs to be, you know, debated. You know, certainly, you know, there should be ongoing discussion about that. But you know, I think that if you look at the references was to, you know, a Chinese vaccine that was being developed with Ad5, and that was the concern. You know, I think that there is a different discussion when one's saying you've got a substantially modified Ad5 that doesn't, you know, as I said, induce those same immune responses. And, you know, is it then the same issues when you're dealing with that? Is it just, you know, is it the exact same vector, or if you modify it and you reduce the immune responses, does that change the question? And I think it's still a point for discussion, but I don't think it's exactly the same discussion that's being had. Yeah, I mean, we got very robust responses back from SAPRA and from our ethics committee at the UCT. And, you know, I think, you know, asking us to justify the use of the Ad5 vector and provide, you know, the data that we provided here today. We were very upfront about these issues and about how we felt that the design of the vaccine, the design of the trial, and the informed consent was mitigating those factors. You know, we in response to that, some of the measures such as you know, doing HIV risk screening prior to enrollment were introduced in response to well, the ethics review from UCT.
Thanks for that. And Tanda's asking, do we have any non-human data on HZ5 and the risk of SIV acquisition?
No, we don't have that data. No. Those experiments haven't been done.
So Graeme, as these discussions evolve, one would imagine that clearly community engagement is absolutely critical, and this particular trial has really been viewed as an important and essential trial to really do thorough, thoughtful and contextual work around engagement. What does that process look like so far, and where do you think more can be done in terms of ensuring that communities are engaged and satisfied? So beyond the traditional CAD structures and those kind of platforms, could you describe that broader civil society and community engagement thus far?
Yeah, look, I mean, maybe Amy can start out with just describing, which is clearly the most important level, is the engagement with the community in Khayelitsha. So, I mean,
yeah, hi. So yeah, I mean, with every study, we first of all go through our community advisory group and present them the study. And really, we went out of our way to make it explicit as to why the engagement around this issue should be of high priority. We've also engaged with Khayelitsha, just to say that there's particular interest around HIV in the study and that we would like to engage on. And we're waiting to hear back from them, but they did sound keen to engage with us. And then again, we're here with you guys as an opening to we're very keen to openly engage around this issue with the community at large and activists.
Absolutely.
Yeah, I would just add, I mean, I think, today is an example of our enthusiasm and great desire to engage in these discussions. And we would be happy to engage with any other organizations that want to enter into have discussions around this. But I think that at this stage, it's the phase 1 trial. It's 20 participants. It's all the measures in place. I think as we go forward, having further engagements about the development process is going to be critical, because there will be other issues that arise, using it potentially in a larger population, how we address that in the informed consent, how you address that in the design, how you monitor for that. And as I've said, the hypothesis that we're going into this trial with is that this vaccine will provide additional protection against SARS-CoV-2, where some deficiencies might occur with the existing vaccines, and at the same time that because this Ad5 vector has been significantly modified from the previous ones, we won't see the increased risk of HIV. Obviously, that's a hypothesis, and we do research to assess, both efficacy and safety in that way, and we need to. But at the same time, protect research participants as much as we can and make them as informed as possible of potential risks, even if we think that those risks are reduced. And so, we will. We're very keen to get a lot of input from broad representatives of community as well as other organizations in the health space. To give us their thoughts and feedbacks about how we negotiate those issues of balancing up. How do you take something that based on the science that exists looks to have efficacy benefits? There might be safety concerns, those might be reduced. How do we assess that? How do we protect participants? How do we explain this in the informed consent process and in the engagement with participants throughout the trial?
Yeah, absolutely. Thanks for that. I mean, it's clear, I think, from the outset that a huge amount of thought and reflection has been put to get us to where we are now, but also recognition of the reason why we make even these initial investments in communications and community engagement is when and if the trial does proceed, it's really setting up a solid ground for the general integrity and perception of the trial. One last question before we wrap up is the issue of, at the core of this, when we were having our preparatory meetings around this issue, a question came up that really struck me to say, what is the communications message? What is the message to communities, especially if this trial proceeds to larger phases, around this modified vector? What information is available that can be taken to communities to, for lack of a better word, prove? Because we're operating in an environment of inherent mistrust, a fragile public health sector, research that has not always been on the side of communities historically. And so really our work, we regard it as an exercise in trust and confidence building. What communications message is there to essentially prove, for lack of a better word, that this vector has been modified and is different from the old one?
Yeah, I think, yeah, Tian, it's a matter of translating the science in a way that that is broadly can be broadly communicated. And I think, to me, some of I think illustrations work very powerfully, and to show that the genetic segments that were deleted in the previous vector, the additional genetic segments, how that changes the conformation of the virus, the proteins that are expressed. That Western blot that I showed you, it does mean that somebody has engaged with science. We need to try and make those messages as clear as possible to people in the community that this is not inducing the same immune responses, and how those logical connections work, because there's that the reason why there was an increased HIV risk, which was in men in the earlier studies, was based on other studies thought to be to the AD5 immune responses. Those were thought the driving of AD5 immune responses is what was thought to increase the HIV risk. This vector has had genetic segments taken out of it. It's therefore expresses fewer proteins, and there is demonstrable evidence that therefore there's less AD5 immunity. And so there's a lot of steps, and we just need to put those in a clearly presented manner, and also just so that this is hypothesis based on the science, because the only way we can ultimately prove this is by doing the research.
Absolutely, and we'll certainly be in touch in the coming weeks in terms of how we can support that kind of communications work. And of course, AD5 is also appearing in some of the other COVID vaccines that are coming up. Is that correct? That we currently have on the radar? Yeah,
so there is a company in China developing an AD5, but that's not a modified one. And so there, perhaps the issues are greater with respect to that. The adenoviral platforms in general are being used obviously quite broadly. So the AD26 has obviously been studied with the Janssen, and but those weren't studied in an HIV context, so we don't have any information on the HIV risk in those. But whether this is exclusively adeno5 or whether this is a broad adenoviral issue, that's something that also needs to be looked at.
Absolutely.
Just on that, AD5, the Sputnik vaccine uses the AD5 and then the AD26 for the boosts, because they are still experiencing that AD5 immune response. So they actually have to change their vector for the boost. And that's not the second generation vector, but that's the other AD5 that I'm aware of.
Thanks, Amy. Thanks.
So, Dr. Ward and Prof. Meintjes, any final words to us as civil society and community groups as we move forward with the small but certainly significant trial over the next few months, amongst all of the noise of the various trials, of the politics of access, of trying to get on top of this pandemic? What is your final message to us for this discussion around how we can support and engage with this very important trial?
Amy, do you want to?
I just wanted to say that, so as a clinician and as a trialist, HIV is my passion. COVID happened, but HIV is my passion. And I want to specifically engage on this issue. And I'm very open to discussions going forward. On the other hand, I'm very excited about this COVID vaccine. We've been working on this since about May last year. And it really is one of the most exciting things that I've, as a clinician, have ever come across. And I really want to just balance those two. And I feel like we have been balancing those two. And I'm going to appreciate the support from the activists in helping us keep that balanced. And my door's always open if you guys want to come and chat.
Thanks, Amy. So yeah, I would just go back to a point that I made earlier, is that, as researchers, we've often faced with this issue where a novel intervention has potential benefits and potential harms. And we need to look at all of the science that exists to the point that we're at currently and weigh those up and make a decision as to whether the potential benefits are great and the potential harms are reduced and mitigated as much as possible. In our view, having looked at all of that data very carefully, is that there is a lot of potential benefits from this vaccine, as I've highlighted in my talk. There are, the changes to the vaccine, are very likely to reduce the risk of Ad5 immunity and therefore the risk of HIV. But it's only by taking this forward and studying this in a responsible way, engaging with community, engaging with participants, being very open about this issue, that we will get those answers that it's of benefit and adds a significant weapon in our armamentarium against COVID without causing increased risk. And as just to echo what Amy said, we're very keen to make this not just a once-off engagement, but to really hear the voice of everybody on this call, everybody from community organizations, to hear your input and help translate that into the messaging that we give to our communities as well as to our participants. So, and just to say thanks very much, Tian, for the opportunity.
Absolutely, thank you. And that's something we will certainly be in touch with. Colleagues, today we spoke to Professor Graeme Meintjes and Dr. Amy Ward, who are leading the ProViva SA1 trial. Thank you again so much for your time, both. Colleagues, next week we speak about COVID-19 vaccines and the search for equity in an unequal world. We will be speaking with Safura Abdul Karim, a public health lawyer and senior researcher at the Wits School of Public Health and a 2020 Aspen New Voices Fellow. She is also a member of the Africa CDC's African Vaccine Delivery Alliance, which aims to support the rollout of COVID-19 vaccines across the continent. She's written on several issues concerning COVID-19 and the law, equitable access to a COVID vaccine, and the effects of criminalizing SARS-CoV-2 transmission on testing efforts and the role of human rights in the COVID-19 pandemic. Thank you everyone again for joining us and spending your Thursday morning with us. We wish you well, and we will see you next week, Thursday. This conversation will continue on africanalliance.org.za, where both the video recording as well as the transcript and Q&A sheet will be published by end of day tomorrow. Thank you again, colleagues, and have a good Thursday further.
Thank you so much.